I'm happy to give you an update on behalf of the team on our efforts to make a small molecule protease inhibitor that targets the SARS-CoV-2 main protease for the treatment of this disease. So we've got a really fantastic team. As the last two speakers were saying, it's really an effort, people willing to contribute their time during the terrible pandemic spring and up to the current time. We've got people from my lab who've been volunteering on the project, from Tom Rovis's lab, who's a professor in the chemistry department. It's been a great partnership with David Ho and his lab and Alex Chavez at the medical center. And then we've had some terrific expertise provided by various facilities around Columbia. Brandon Fowler and the mass spectrometry facility, Farhad Faruhar did all the crystallography, and Chuck Farron, who did the fragment screen, I'll mention briefly. And then we've got some in-kind contribution from Waters Corp, which volunteered their analytical chemistry expertise to help with the project. So we're really grateful to everyone's efforts. We've also gotten some funding from Columbia Technology Ventures, the TRX Accelerator, and the Jack Ma Foundation. So the hypothesis we're working under here is that viral protease inhibitors, direct-acting antiviral protease inhibitors, would be effective treatments for SARS-CoV-2 infection because these were really transformative in other viral infections, HIV protease and HCV protease, for example. And that introduction of protease inhibitors is really the turning point in the treatment of HIV. So just to get everyone on the same page, so the SARS-CoV-2 viral RNA genome encodes two large polyproteins that have to be cleaved at least 11 different sites by what's called the main protease, or I'll refer to it as the 3CL protease. The polyprotein then gets broken down into smaller proteins that are involved in necessary for the replication of the virus. So if we can block this cleavage event, then we should have an effective direct-acting antiviral treatment. And the exciting thing here is that the main proteases are very similar across the different coronaviruses that have been reported to date. So if we had a potent drug-like treatment that was able to inhibit the main or 3CL protease for SARS-CoV-2, it's very likely that it would also inhibit other coronavirus proteases and therefore be a broadly effective treatment for coronaviruses, including any future pandemic coronaviruses. So the question then is it possible to inhibit the SARS-CoV-2 3CL or main protease with a drug-like small molecule? And so we took a broad approach to this question. We tried repurposing compounds that had been reported for other proteases and SARS-1 protease, other viral proteases. We also did a screen of more than 7,000 electrophile fragments. So those are very simple compounds that could be starting points, give us some ideas of what kind of electrophiles can bind to the protease. And then we did a DNA encoded library screen with more than 3 billion compounds. And so a number of these are still progressing through our pipeline and will probably yield some additional scaffolds that could be optimized and developed. But I'm going to tell you today about the most advanced scaffold that we've been working with. So actually it's based on GC376, which has been studied for the treatment of cat coronavirus infections. It's a peptidic compound, relatively peptidic, and it has this sulfonate warhead that gets unmasked to make an aldehyde, and that generates a reversible but covalent inhibitor of the protease. And this was effective at reducing viral load in a kind of clinical, or what we would call a preclinical study of cats that were infected with the feline coronavirus. So this was one of the compounds that seemed quite promising in our early testing, where we found that if we could see the binding by biophysical methods such as calorimetry, we could get a submicromolar KD. out from those analyses, we could see inhibition in a biochemical assay where we measure the protease activity in an in vitro system, and we see an IC50 in that assay about 100 nanomolar. And then we've been using the Vero E6 cell model with the SARS-CoV-2 genuine virus in David Ho's lab, and the potency in this assay is in the low micromolar range, about 13 micromolar for GC376. Those cells have a lot of efflux, drug efflux mechanisms, so we think that's artificially higher than it would be in cell models that don't have that efflux. So we're trying to test that now, but we do see the antiviral activity even in that high efflux cell system. And the good thing about this scaffold is that in contrast to many others, it has a good plasma and microsomal stability, as shown here. So this is reflective of liver metabolism and metabolism in plasma. So it looks like it's a stable scaffold that might be suitable for development as a therapeutic. So one of the questions we had was, does this compound and do any of these compounds make specific interactions, specific one-to-one complexes with the protease? There have been a bunch of compounds reported in other papers in the literature that some of those we find are relatively non-specific. They bind at multiple sites. They may be more promiscuous. So we can see the specific interaction here in several different ways. For example, we can look by mass spectrometry at the intact protein and see a shift in the mass when this compound binds. Now, this is a different compound, what we call compound 4, which is also a covalent inhibitor. And then we can compete that off with the GC376 scaffold. In this assay, GC376 is reversible, so we can't see the complex, but we can see the competition with the adduct made by this other compound. And then we also developed a way to detect with native mass spectrometry the complex, the direct complex of GC376. The protease is a dimer, so then we should get 2 GC376 molecules bound, and we do see that. So those with some other data convince us that GC376 is making a nice one-to-one complex. It's binding in the active site, and it's binding to the active site cysteine, as we would expect based on that aldehyde that gets unmasked. Of course, we want to test for selectivity of these scaffolds against the viral protease versus other proteases. If compounds were broadly active against human proteases, that might cause some undesired toxicity. So we set up a selectivity test using 2 human proteins, human caspase-3, which is a cysteine protease like the SARS-CoV-2 main protease. And with the caspase-3 assay, we see at a 600-fold selectivity with GC376 itself, so it's very selective. And then we also tested against chymotrypsin, it's a human serine protease, and there we don't see any inhibitory activity on chymotrypsin for GC376. So it looks like it's a good, the scaffold has good selectivity, at least against these 2 human proteins that we're starting with. And then we've made a series of analogs now around this scaffold to see how amenable it is to improving the potency. And so first we screen all these at a high concentration, and at 50 micromolar, we see complete inhibition of protease activity. And then we test them in a dose response series, and we're able to get compounds that are more than 20-fold potent compared to GC376 now again, in the biochemical protease assay. So some of these are now down in the single-digit nanomolar potency range. For example, this is one of the more potent analogs. We've looked at ED34, which has an 8 nanomolar IC50 in the biochemical assay. Actually, the most potent analog we've seen today just in the last few days is about 3 nanomolar. So we're getting close to potentially breaking the picomolar barrier, which we think would be sufficient to have a lead that could then be further developed if all the other properties were suitable. We tested these some of these more potent analogs now in the selectivity assay. So for caspase-3, for example, these more potent activity analogs have no activity on caspase-3, unlike GC376, which I said has some activity but still very selective. So these compounds are very selective so far for the viral protease. And against chymotrypsin, the same story. We don't see any inhibitory activity against chymotrypsin. So these compounds look, again, to be like good selective antiviral compounds. We've put some of these also through the same battery of assays I showed you for GC376. And we can see, again, a nice complex between these compounds and the protease. A one-to-one complex. Looks like they're binding in the active site as expected. So again, here's the assay where compound 4 shows the mass shift. And then we do a competition with these more potent analogs. And again, we can compete off the binding of compound 4, which binds in the active site to the active cysteine, suggesting that these compounds are also binding in that site. We've also been able to obtain, with Farhad's help, crystal structures of many of these more potent analogs found in the active site, so we can see the binding mechanism, orientation, the kind of interactions that are made. And we're using that information to try to design the most potent analogs that we can. Now, we've tested these with David Ho and Shou in his lab against the genuine SARS-CoV-2 virus. And again, in the Vero E6 cells with the high efflux, they have activity in the low micromolar range. We believe that as we shift to other cell models, we'll be able to see potencies that are more aligned with the biochemical assay. But to date, these compounds do have some improvements over GC376, even in the Vero E6 cell assay in the kind of 1 to 5 micromolar potency range in that assay. And then Alex Chavez's lab has set up a nice assay to look for the broad activity against other coronavirus proteases. So in this system, they express different viral proteases in human cells, which results in cell lethality. And then if you can prevent that cell lethality, you can see the antiviral or protease activity, inhibitory activity of the compound. So we can see GC376 and these more potent analogs shown here all have the ability to inhibit the SARS-CoV-2 activity in this assay, as well as original SARS-1 and MERS protease activity. So it looks like, as we hope, these compounds have, the scaffold has broad coronavirus antiviral activity, and that these should be suitable potentially for other coronavirus infections in the future.
Brent, sorry to interrupt. This is a 2-minute warning.
Perfect. Okay, it's my last slide. So to summarize where we are, the SARS-CoV-2 3CL or main protease is amenable to potent inhibition with small molecules, and other groups are pursuing this as well and finding the same thing. The ADME properties of this scaffold we believe are promising, but we're exploring other scaffolds as well. This protein is very amenable to X-ray crystallography, so we've gotten lots of great structures, and we think that's really helpful in designing the most potent compounds that we can get. And so the next steps for us are to evaluate the ADME properties, the kind of stability and pharmacokinetics of these compounds, and further enhance the potency a bit. And then if we can identify a suitable lead from these studies, begin to do IND-enabling safety studies to see if we have a compound that could be advanced into clinical studies. And this would be potentially for early treatment of SARS-CoV-2 infection, and as a kind of additional tool in the arsenal of treatments beyond vaccines or biologics that are obviously being pursued by many groups. So I'll stop there and take any questions.
Thank you, Brent. If I can start again with a simple question, and that is, for your most potent inhibitor, I think the one you mentioned with a 3 nanomolar IC50 in the in vitro assay, is it toxic to cells?
We don't see toxicity in cells. Yeah, exactly. In fact, it's the opposite, that in Alex Chavez's protease activity assay, where expressing the viral protease is lethal to cells, you rescue that toxicity. So at least in the concentration ranges we're looking at, we don't see cell toxicity.
Okay. And another naive question, maybe you answered this and I missed it. If you compare the active sites of the proteases from the different coronaviruses, in particular, you know the amino acids that the scaffold is contacting, correct? Are those highly conserved across the coronaviruses?
They're highly conserved. They're not exactly identical, but they're highly conserved. So looking at those interactions and the structures of the different coronavirus proteases, it seems very likely that you would have broad activity against most, if not all, coronaviruses for these compounds.
Okay, and we have a question posted in the Q&A box from Dong An. Dong, if you could raise your hand, it might be better if you ask your question. So Dong, you can talk.
Yeah, thank you, Professor Stockwell. I'm very enjoy your talk. I have maybe naive questions. I saw you show that when the free CL and the caspase-free treated by GC376, or maybe I'm wrong. They showed different IC50. Even they use cysteine residues to decompose their target protein. So I'm wondering what caused the different reaction between them.
Yes, it's a good question. I didn't go into the details, but basically there is a covalent interaction with the cysteine, but there are also many non-covalent contacts that drive the binding. So, and those contacts we see are important because as we modulate those with different analogs, you get big changes in the potency. So you need, like many covalent inhibitors, you need first the non-covalent contacts that drive the binding, and then once the cysteine is positioned near the aldehyde, it locks in to make that reversible covalent interaction. And you don't have those non-covalent contacts with caspase-3, even though it has a cysteine in its active site.